Hello, good afternoon, year 13. Year 13 now, right? Not year 12. How are you doing today? Let me know who's here. I can't see any of you in the chat yet, so do say hello so I know who is here for our Friday afternoon lesson. Hi Grace. How are you? Hi Jennifer. I'm so glad you've come back. I thought maybe I'd scared you off year 13 biology yesterday with our difficult lesson on PCR. Good to see some of you have come back for more, even though it's sunny outside, or it is here anyway. Okay, so yesterday we kind of did a lesson where we revised DNA replication and then we learned something new. So we did replication in vitro, or PCR, the polymerase chain reaction. We're going to do something similar today. So we're going to do transcription, and we can revise translation as well if you want to. And then we're going to move on and look at something extra for year 13. Okay, does that sound okay? Put a plus in the chat if you're happy with that. If we want to spend longer revising transcription and translation, we can do that. But once I know you're confident with those processes, ideally, I want to introduce something new and get a little bit ahead with our year 13. Sienna says, yes, please. That sounds good. I love the positivity, Sienna. So is anyone new, by the way? Was anyone not here yesterday? Because I've got this slide again, like I meant to introduce myself, but if no one's new, there doesn't seem very much point doing that. This is me. Hey guys. Oh, so is it Somi or Somi? Somi is new. So my name is Laura. I'm a biology teacher. I don't really need to say anything else, right? I've got a lot of experience of teaching A-level biology. I have done a lot of work with exam boards, so hopefully you're going to find these lessons really useful to prepare you for your exams, which will come around faster than we know. Right, should we just get straight into it? Transcription. Now we're going to have to switch our brains a little bit because yesterday it was all about DNA replication and today is all about transcription, which some students do confuse, right? Because there's a few similarities between these processes. So a simple question to start with, where in the cell does transcription happen? Where are we? Let's get inside the cell. Where are we, guys? Where does this happen? Oh, we've got a couple of different answers. So some of you are saying ribosomes and some of you are saying nucleus. What do we think is correct? Remember, this is the first stage of protein synthesis, not the second stage. So at this point, we're not actually making the polypeptide, okay? So those of you that said nucleus are correct, okay? Because this is stage 1 of protein synthesis. Okay, so we're in the nucleus, so let's get inside the nucleus. So what are we making? This is the next blank. Something is produced using the DNA template strand. What are we making? I mean, if you look at the diagram, there's a bit of a clue. Well done. What does the M stand for? What does the M stand for? That's an easy question. Just warming you guys up. Messenger. Thanks, M. Thanks, Grace. Tempered steel. Yeah, we're making mRNA, right? So one of the things to remember, just to save any confusion with DNA replication, we are not making DNA, we are making RNA, messenger RNA. So only one strand of the DNA acts as a template. Have a look at the diagram. We've got the DNA, which has been separated. We can see the DNA backbones here in blue. Yeah, so we've got our 2 DNA backbones. We've got our exposed bases. Only one of those strands is acting as a template to actually build the new mRNA, okay? My next question then for you, which enzyme, or enzymes, if you like, are involved in the process of transcription? There's one that you have to be able to name, but you might give me 2. Which enzyme or enzymes do we know for transcription? Ooh, okay, thanks, Jay. Yes, tempered steel, good. Sienna, have a careful think about your answer. Remember, we're making RNA. So have a little think about that. Grace, again, we're making RNA, so have a little think. We've just started, don't worry, Saya, you've not missed anything really. So, okay, we can name DNA helicase to start with. Quite often on mark schemes, they don't give you credit for this enzyme, but we can name it if you want to go back that far. DNA helicase, what's the job of DNA helicase? The same as what it does in DNA replication. What is the job of that enzyme? Because it's the same job here, right? And then we'll get on to our second enzyme. Thanks. So between you, you've given me both of those answers there. So we've said DNA helicase breaks hydrogen bonds and separates the two strands of DNA. Okay, which we can see on the diagram has already happened. We can see that the DNA strands have been separated. So one strand acts as a template. So it's a little bit different to DNA replication in that sense, because in DNA replication, both strands are acting as templates. So one strand acts as a template. What is the next thing we could say before we start talking about the next enzyme? Okay, so we've used our DNA helicase, we've broken the hydrogen bonds, we've separated the two strands, we've got our exposed bases on the template strand. What's going to start to happen? Any ideas before we talk about the next enzyme? Oh, good, Grace. Grace has given us an example of some base pair matching. Okay, and you've remembered we're making RNA, so Grace has actually said uracil. Good, Ella. Okay, so nucleotides line up with, I'm going to say, the exposed bases on the template strand. You can see that on the diagram. Okay, now sometimes you actually get a mark for giving examples of this base pairing, of the base pair rule. So we could go ahead and give examples. For example, if there is a T on the DNA, because there's no thymine in RNA, right? But if there's a T on the DNA, that would pair with an A on the RNA. I'm going to ask you to do the others for me. So if there's an A on the DNA, what would that pair with in the RNA? Think carefully. If there's a C on the DNA, what would that pair with? And if there's a G on the DNA, what would that pair with? Good, Sienna, Daisy. Remember M, we're making RNA, so we've got RNA nucleotides moving in. There is no thymine in RNA. Good, so if there's an A on the DNA, that would pair with a U base or a uracil on an RNA nucleotide. The C, good, would pair with a guanine, and the guanine on DNA would pair with a cytosine on RNA. Okay? So A says, is it RNA helicase or DNA helicase that separates the two strands? It would be DNA helicase, because remember, we're separating the DNA strands at the start of transcription. So we're breaking the bonds in the DNA and separating the DNA strands so that one of them can act as a template, right? So that would be DNA helicase. But we're about to move on to the second enzyme. Now, a lot of you have already given me the name of this enzyme. Once those nucleotides have moved in and they've followed that base pair rule, that complementary base pair rule, we're going to move on to our second enzyme, which is the most important one. This is the one that we have to be able to name. RNA polymerase. Okay, and it's RNA polymerase because we're making RNA. Perfect. Right, tell me the job. What is the job of RNA polymerase? It's very similar to the job of DNA polymerase, but obviously this is RNA polymerase. So what is the job? Oh, Sienna, love it. Perfect. You've got like 2 marks there. Well done, Grace. So it joins the nucleotides together. I'm going to put in RNA nucleotides just so we don't forget that we're making RNA. So it joins the RNA nucleotides together, and you can say adjacent nucleotides because someone said that in the chat. So it joins RNA nucleotides together, but we always name the bond, right? So we say by making, and this was well remembered, by making phosphodiester bonds. Perfect. Perfect. How come 2 strands don't act as a template? You only need 1 strand to act as a template because you're just making a single-stranded piece of messenger RNA. So look at the diagram. All you need is 1 template strand. And we're making single-stranded, a single strand of messenger RNA. And obviously the bases on that messenger RNA, or the order of bases on that messenger RNA, are determined by the order of bases on that template strand. In fact, that can sometimes get you a mark for this kind of question, just for saying, let's write it down, the order of bases on mRNA is determined by the order of bases on the DNA. We could even say on the DNA template strand. Okay. Will A pair with T occur in transcription or just AU? So if there's a T on the DNA, Jennifer, that would pair with an A on the RNA, but you're never going to have a T in RNA. Yeah, but if there's a T on the DNA, that would pair with an A on the RNA. Okay. Right, so we've kind of finished the process of transcription at this point. Where is this RNA going to go, guys? Because this was happening in the nucleus. So once that RNA is complete, where is it going to go? Good, to the ribosomes. What does it have to leave through? I'm just trying to get all of your knowledge out here. What does it have to leave through? Grace is saying out of the nucleus. How does it get out of the nucleus? Through what? Thanks, Daisy. It's going to go out through a nuclear pore. Loving your answers today, guys. It's going to go out of the nucleus through a nuclear pore, which is in the nuclear envelope. Well done, Jasmine. And it's going to go to the ribosomes. Good answer there. Johan's given us a full answer. Through the pores, the nuclear pores, to the ribosomes. Okay. How do we feel about this? Because I can spend longer on it if you want to. Give me a little score maybe in the chat. I think we did this yesterday. Who's on like a 9 or 10 confidence level? Who is on like a 1 or 2 confidence level? Let me know, because I can kind of judge then. Oh, we've got some high numbers. Okay. This bodes well. If you're on like a 4 or a 6, have you got any questions about this then for me? Some of you are giving me really high scores. Some of you are saying, oh, I'm not really 100%. So have we got any questions? Is there anything specific? Oh, Sienna's saying 9 out of 10 for transcription, but a 4 out of 10 for translation. Does splicing happen in the nucleus? Yes. So I've not actually mentioned splicing here, but well remembered, Ella. So when the mRNA is produced, it does still contain introns, right? Yeah, pre-mRNA or primary mRNA. It does still contain introns. So they do need to be spliced, yeah, which happens in the nucleus. So yeah, we could mention that as well. I wasn't going to go into that bit today, but yeah, splicing happens in the nucleus. We're going to remove the introns and then we have our mature messenger RNA. How do we remember what kind of bonds are broken and formed? Good question. Don't really have tricks for you. except if it's a polymerase enzyme, like DNA polymerase makes phosphodiester bonds, RNA polymerase makes phosphodiester bonds. You kind of just have to remember that. And then helicase always breaks hydrogen bonds, right? DNA helicase always breaks hydrogen bonds. So you just kind of have to learn it. It's just repetition, isn't it? Keep revising it. Does the mRNA have paired bases? Good question. No, it's single-stranded. Okay, have a look. Can you see here? Have a look at our mRNA. Can you see these are unpaired bases? It's single-stranded. H is for helicase and H is for hydrogen. I love it. Is splicing considered to be a part of translation? No. It's not really part of transcription or translation. It's after transcription and it's before translation. You could include it on a question about transcription. You know, you could stick at the end and then splicing occurs and the introns are removed. But usually you'd have a separate question about splicing because it's after transcription and before translation. Okay. Yeah, Jasmine, you're not wrong. Quite often you don't mention DNA helicase in transcription. You can do, but the enzyme that they are going to give you marks for in transcription is RNA polymerase. That's the one they want you to know, okay? The job of RNA polymerase and what that does. Is splicing in OCR? Yes, it is. In year 13, though, for OCR, not in year 12. Whereas for AQA, splicing is in year 12, so you would have already learnt it. But Jay, if you're doing OCR, you probably just haven't learned about splicing yet, but it is coming up for you in OCR year 13. You do it towards the end of year 13. Okay. Right. Let's move on. What is this? I've left the title blank. I don't even think you've got the title in your workbook, but I thought I'd throw this in while we're doing transcription. This is the process of what? What is the process shown by this diagram? Translation. Good. Let's put this in. Second stage of protein synthesis. Okay. And you all know where this happens. You can put it in the chat again, though, for me. We all know where this happens because we've just said, haven't we, that the mRNA leaves the nucleus through the nuclear pore, goes to the... Exactly. So we're going to fill this in. Happens at the ribosomes. Now, the whole point of translation is to actually make the polypeptide or the protein chain, yeah? So now we're going to say the sequence of bases in the mRNA, which goes to the ribosome, determines the sequence of what in the polypeptide. Or we could say protein probably there. Get away with it. Well done. Good work, Grace, J, Swaro. Amino acids, okay? So the sequence of bases in DNA determined the sequence of bases on the mRNA. And now the sequence of bases on the mRNA is going to determine the sequence of amino acids in the polypeptide, which is just the chain of amino acids, right? That forms the protein. Okay. Shall we try and describe this process? Can anyone... I mean, what things do we know? I want to know what you remember. If you were given a 5-mark question right now on translation, describe the process of translation, are there any bullet points that we could throw down? Is there anything we can recall? Not even in the right order. Let's just see what you know. So Jay is saying it's something to do with tRNA. Yes. Jasmine is saying, I thought tRNA determined the amino acid because it had the amino acid. You're not wrong that the tRNA is definitely carrying the amino acid and they are carrying specific amino acids, but the binding of the tRNA is determined by what's on the mRNA, right? So this is the kind of key phrase that we use. The sequence of bases on the mRNA determines the sequence of amino acids. We've got some good statements coming out here. Codons on the mRNA are complementary to the anticodon on the tRNA. They match up. Good. Yes. tRNA has an anticodon complementary to the codon and then it attaches. Nice. Nice. And Jay is saying the tRNA brings the amino acids. Got some good, I think as a group here, you've pretty much got the full story. Amino acids join together. Oh, Jasmine, yeah. Peptide bonds form between amino acids with ATP. Guys, I think you've pretty much just done the whole story between you. Should we do it in a nice order though? So I'm going to start off by saying the ribosome moves along the mRNA. So, I mean, I could have even said the ribosome like attaches. Remember, the ribosome has 2 subunits and it kind of goes around the mRNA. The mRNA is kind of like threaded through the center. So you could even say the ribosome like attaches to the mRNA. The ribosome moves along the mRNA. And then I'm going to use some of your statements. So tRNAs carry specific, it's really important that we use the word specific amino acids to the ribosome. And I think Jasmine said before that the tRNAs have, can you see here? They have what's called the amino acid binding site. Yeah. So, but they're specific. They carry specific amino acids. It's not like any tRNA can carry any amino acid. Each one carries a specific amino acid, yeah, which is determined by the shape of that binding site. This is kind of like a 6 marker, yeah. This is what I would write for a 6 marker. Obviously, with 5 and 6 markers, there's sometimes alternate marks that you can get. There might be 7 or 8 marking points, but yeah, I'm trying to put together like a decent 5 or 6 marker. So we've got ribosome moves along the mRNA. The tRNAs carry specific amino acids to the ribosome. And then the anticodon, which is the exposed 3 bases, which you can see here, for example. The anticodon on the tRNA will bind to a complementary codon on the mRNA. Have a look at that happening. Have a look at this here. We've got a tRNA carrying a specific amino acid, which is lysine on this example. You can see the anticodon, UUU, binding to the codon on the mRNA, which is AAA. Okay. Only 2 transfer RNAs at a time. Well remembered. That's true. You might even get a mark for saying that. Yeah, 2 transfer RNAs can fit in at a time. I'm just pausing because I'm looking at my diagram and I'm thinking, this shows 3, but that is incorrect. Because you're right, Jennifer. It is actually only 2. So I might have to get this diagram edited. That is a good spot because it is only 2. So the anticodon on the tRNA binds to the complementary codon on the mRNA. I'm going to steal Jennifer's point and say 2 tRNA can fit in ribosome at one time. Okay, not 3 like the diagram suggests. So once the tRNAs are both in there, 2 of them, what's going to happen between the amino acids? Because they've both carried an amino acid. What is going to happen? I think some of you said this before in the chat, but let's repeat it. Once we've got those 2 amino acids in the ribosome, what is going to happen? Well done, Grace. What kind of reaction is that as well? What kind of reaction if we're making a peptide bond? Well done, guys. Condensation, you're right. So the peptide bond forms between the 2 amino acids. It does use ATP, which might pick you up an extra mark. And you might be able to get an extra mark for saying this is a condensation reaction, but not always. It depends on the mark scheme. Running out of space to write, aren't I? Okay. And I'm going to say this process continues. Until when? When will this stop? Because we're going to keep having tRNA carrying specific amino acid, moving into the ribosome, anticodon binds with codon. This process continues, good, until a stop codon is reached. I think we've just about fitted a nice answer in there. Okay, is there anything that I've missed? Anything else that your teachers have told you that you're questioning whether you can pick up marks for? But yeah, I think I would probably leave my answer there if it was a 5-6 marker. I think I've given enough detail. Obviously, once the stop codon is reached, then the polypeptide is complete, right? It gets released. Why can only 2 tRNA fit in a ribosome at the same time? I'm guessing it's just to do with the size and the shape of the ribosome. There are like 2 tRNA sites inside the ribosome. You didn't learn stop codon. Oh, that's interesting. I think for OCRA, even in year 12, you should have learned stop codon. So the stop codon is a specific sequence of bases on the mRNA, which basically marks the end of the polypeptide. Like there's no amino acid for that codon. So the polypeptide is basically completed and it gets released from the ribosomes. Okay. Okay. 5' to 3'. In what context, Zainab? Are you thinking about DNA replication? Or is it because it's on this diagram? Because I don't think I'd ever mention 5' to 3', to be honest, on a translation question. But it's to do with the carbon number. So, like on this mRNA is obviously single-stranded, so I only need to draw one strand. But this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4. Carbon 5 is on this branch, okay, because this is ribose sugar and it's got an oxygen at the top. So this end would be described as the 5' end because it's the end that carbon 5 is on, whereas this end would be described as the 3' end because it's the end that carbon 3 is on. So that's kind of how we differentiate. The 5' has the 5 carbon end and the 3' is the 3 carbon end. I don't think I would ever use it. I'd never seen it come up with translation. I know it's marked on this diagram, but okay. Okay, right. And some of you are talking about sense and antisense strand. Again, I think you're slightly overcomplicating it here. When we're talking about transcription, you can just call the DNA strand that is being used to make the mRNA or is acting as a template, we tend to just call it the template strand. Some of you are throwing all sorts of leading strands, lagging strands, all these things. Okazaki fragments, yeah, you definitely don't need to know this unless you're doing some different exam board that I am not familiar with. I don't know, you'd have to tell me who's just talked about the Okazaki fragments. I think I learnt about those when I did my A-level, but it's not on OCR or AQA. So we can ignore that unless you're doing a crazy exam board that's really, really hard. Yeah, I think some teachers do go a little bit beyond spec, don't they? If they're maybe really interested in the topic or they're looking at some old spec questions, they might throw some extra stuff in there, but you don't have to know those extra bits. Okay, right. So now we've revised transcription and translation, which was year 12. I thought that we could move on and think of something a little bit ahead for our year 13 Year 13 content. So I'm going to introduce something new to you that I don't think any of you will have done in year 12. Okay. Are we ready? Oh, I'd forgotten about this. Before we do that, ignore me. This is a nice activity. This is just labeling stuff. So let's do this first because this is a good little checker. Everything is numbered here. I want you to tell me what you think that number is pointing to. So put the number in the chat and then give me the label. I'm just going to edit this diagram a little bit because 10 is meant to be an organelle. You're going to name it for me. Okay. So let's do this first and then we'll move on to the new stuff, right? We've got numbers 1 to 10. Some of them are a little bit ambiguous, but I suppose that just means there's a couple of options you could give me that would be correct. And we'll see how we get on. Yeah, I think there's a few that you could argue a couple of different things for. Okay, so cell membrane, or always better to say cell surface membrane if you can, or you could call it the plasma membrane of the cell. 2, good, the cytoplasm. 3, good, is the DNA. 4, very impressive. This isn't like an exam question, by the way. This is just a diagram that I've put together to try and get you to recall these processes and the key terms. So 5 is, yeah, transcription. Should we call 5 transcription? It's like the process of transcription being shown there, isn't it? 6, yeah, I would agree. That is our single-stranded mRNA. 7, what is 7? Is 7 pointing to this kind of arrow bit here? Maybe it's meant to be a nuclear pore, or you could say it's like the mRNA leaving through a nuclear pore. Maybe, because it's kind of got an arrow coming out there, hasn't it, for number 7? Number 8. Can it be nuclear membrane? Yeah, it is a nuclear membrane. Yeah, it's a double membrane, nuclear membrane. We just tend to call it nuclear envelope. Yeah, but nuclear membrane is fine. 8 is our tRNA, which, oh, what does T stand for? I didn't ask you that one. What does T stand for in tRNA? Number 9. Are we just saying translation? I was going to say, what was I going to say for number 9? Yeah, so the T stands for transfer. A few of you saying transport or template. It actually stands for transfer. I suppose because it is transferring the amino acid, right? Number 9, I think there's a few different things we could say. We could say it's amino acids. We could say it's the polypeptide. A few of you have just said it's the process of translation, which I suppose is fine. Yeah, you can actually see number 9, you can kind of see the amino acids being joined together. You can see the polypeptide being formed. So it's part of the process of translation. Okay, number 10 is the one that you're probably not going to get because the diagram went a bit dodgy, but it's meant to be the ribosome. So it's meant to be like a circular structure. It's meant to be the organelle, the ribosome in which this is taking place. And then what is 11? I can't see any answers for that one. Can someone give me the answer for number 11? I think it's probably gone off of my chat. What are we learning about? So we're just recapping year 12 protein synthesis. So we're learning about transcription and translation. And this is like an overview of the whole thing, right? Yeah, thanks, Sienna. So 11 is the mRNA. Again, can you see single-stranded mRNA? You can see here, look, the anticodon on the tRNA is binding to the codon on the mRNA. So they're complementary base sequences. You can see that single-stranded mRNA. So remember, we say the order of bases on the mRNA is ultimately determining the order that those amino acids will get put together in. Okay, nice. Okay, hopefully you feel a little bit more confident now on those topics, a little bit of year 12 revision. We are gonna look at something new, okay? So this is what you didn't learn about in year 12. I feel like a lot with biology. I remember like feeling when I started doing A-level that my GCSE teachers had lied to me because they started telling me all this new stuff. And I was like, no one told me about this at GCSE. It's kind of like that here. It's like when you were learning about transcription in year 12, no one told you about regulation of transcription. But I guess we're trying to build the difficulty, yeah, through year 12 and year 13. So, as you know, or hopefully you know, not all genes in a cell are expressed. So, even though all of your cells have the same genes, they are not all being transcribed and translated. Like, all of your cells have got the gene for insulin, but is it being transcribed and translated in all of your cells? No. Can anyone tell me where insulin is transcribed and translated? Which type of cell is insulin actually produced in? Exactly. It's in the pancreas, cells of the pancreas. But like all of your other cells have that gene, right? It's just not being transcribed and it's not being translated. So the cell is not producing that protein, okay? So that's what we're starting to think about here. So the transcription of genes is controlled by transcription factors. Now, I've given you a definition here of a transcription factor. So the first thing to remember is they are proteins, because you could be asked, you know, what are they made from? So they are proteins. And what they do is they bind to the DNA at a site called the promoter region. Well done, Ink. Yes. Ink's just said insulin is made by the beta cells of the pancreas. Well done. I didn't think you'd know that yet. So transcription factors are proteins. They bind to DNA. So these are key pieces of information, okay? And where do they bind to DNA? At a region called the promoter region. Now, transcription factors can either initiate transcription, cause transcription, or they can inhibit transcription, stop transcription. Okay, yes, it is the Langerhans, the islets of Langerhans. So we've got two types of transcription factors. We only really learn about one type in A-level biology, which is the activators. So we only really learn about the transcription factors that switch on gene expression, okay? They initiate transcription and therefore cause gene expression. But just be aware that there are also transcription factors that act as repressors. They actually stop transcription. They turn off gene expression. What AQA topic is this? It's the start of module 8. Control of gene expression. I'm going to say it's the start of module 8. I'm going to say that. I think that's right. It's the start of module 8 for AQA Sienna. It's towards the back end of year 13. And for OCRA, you're testing me now. I can't remember the modules in OCR, but it's towards the back end of year 13. You're going to have to get your specs out and have a little look. It's towards the back. It's when you're doing like genomes and gene expression. Oh, cellular control. It's called cellular control for OCR. Yeah, cellular control, regulation of transcription in cellular control. And I'm going to trust what Somi says, genome projects for AQA. Okay, why is this done? It's exactly so we can switch genes on and switch genes off, because not all genes want to make all proteins, right? In some cells, you don't want to make that protein, but in other cells, you want to make lots of that protein. So we can regulate transcription to control how much protein they make, whether they make that protein or not. Is that gene on or off? Okay, thanks, Em. Em's saying topic 8.2 for AQA. Nice one. Yeah, I knew it was the start of module 8. Okay, so let's have a look at how this actually works. Is this done by humans? Yeah, this is happening in all of our cells. What's the promoter region like? We're going to look at that now. So a bit of protein revision, yeah? Transcription factors have a specific shape. They are proteins. What do we know about proteins from year 12? They have a specific tertiary structure. So a transcription factor is a protein. So it's all about shape and structure. Guys, can you tell me which bonds hold the tertiary structure together? Yes, the 3D shape. These are globular proteins. Which bonds are going to hold the tertiary structure of these proteins together? Good one. The tertiary structure, not the primary structure, Swaro. So Swaro said peptide bonds, which obviously are in proteins, but that holds the primary structure together. It just holds the amino acids in order. But for the tertiary structure, we should be able to name hydrogen bonds, disulfide bonds, or you might have called them disulfide bridges or sulfur bridges. And then we've also got ionic bonds. That's just year 12 revision, right? Someone said covalent. You are correct. Disulfide bonds are covalent bonds. So they're really strong. Okay, nice one. So they're proteins. We know proteins have a specific tertiary structure. It's very complex. They bind to DNA at a specific base sequence called the promoter region. Now, someone asked what the promoter region was. So it's basically a region, I'm going to put of DNA, found at the start of each gene. So if you have a look at my diagram, if we imagine that this is the gene, okay, sequence of bases that codes for a polypeptide, at the start of that gene, can you see highlighted in blue, is the promoter region. So it's a specific sequence of bases at the start of the gene. Now what happens is the transcription factor, here's my transcription factor, the transcription factor can bind to that promoter region, okay? Now remember, the transcription factor is a protein, so we can start to think about shapes fitting together. Transcription factor is complementary in shape to the specific base sequence in the promoter region. Okay, because it's got a specific tertiary structure given by, you know, where its bonds form and all that kind of stuff. But the shape of the transcription factor has to be complementary to that promoter region. It's going to bind to that promoter region when those shapes fit together. Okay. But look what happens. When a transcription factor binds to the promoter region, it will allow RNA polymerase to bind, and that will initiate transcription. Okay, so let's simplify that. I feel like I want to just do a little simplified version of notes. So transcription factor binds to promoter region. Okay, this allows RNA polymerase to bind. And this is ultimately going to initiate transcription. So we've switched on gene expression. Can you see that? This is an activating transcription factor. If it binds to the promoter region, then RNA polymerase can bind, then it can cause transcription. then we're going to make that protein. Yes, exactly. This is happening in the nucleus. So this is before transcription, which we started the lesson with. Before transcription, or so transcription can occur, we actually need a transcription factor to bind to the promoter region. Then RNA polymerase can bind. Only then are we going to get transcription, okay? And we're going to get gene expression. Hopefully this is making sense. Okay. Just because I'm cautious of time, I think I'm going to, this happens, yeah, this is to do with transcription. So this whole topic is basically control of transcription. Okay, so what I want you to take away from this, obviously we're not going to be able to cover everything, but what I want you to take away from this is what you didn't learn in year 12 is that for transcription to occur, you need a transcription factor to bind first. So the transcription factor binds to the promoter region, which is the start of the gene, then RNA polymerase can bind, then we initiate transcription. Okay, so if that transcription factor is not produced in that cell, you're not going to get transcription of that gene. You need that transcription factor in order to initiate transcription. Okay, so if the cell is not making that transcription factor, then that particular gene is not going to be expressed in that cell. Okay. Chat's gone quiet. How does it get produced? You tell me. What did we say a transcription factor was made from? It's a really good question. Nafisa just said, how does the transcription factor get produced? Daisy said the transcription factor is a protein. So where does it get produced? So yeah, Saya says transcription factor is a protein. So where does it get produced? Where are proteins made? In the ribosomes. Exactly. So to make, this is one of those chicken egg situations, right? The transcription factor is a protein. So to make the transcription factor, you need a gene that codes for the transcription factor. You need to transcribe it and then translate it at the ribosomes. And then the transcription factor can switch on transcription of other genes. So which, I'm confusing myself now. Do you see what I'm saying? I'm like, hang on. If you need a transcription factor to turn on transcription, where's the transcription factor that turned on transcription of the gene that coded for the transcription factor? One of them biology questions. I can't answer it. How does that work exactly? Because there are genes that code for transcription factors, and then the transcription factors get produced and then they switch on other genes. It happens in science. That's going to be the answer, isn't it? It's just science. It's like magic. Who knows? Okay, I bet someone could answer. Someone, like some university professor is going to answer. Okay. I've clearly overplanned for today's lesson, and this stuff is really quite hard. So I'm quite conscious of time. What I think I'm going to do is I'm going to skip to some of the questions so we can just check what we've actually learned so far. I don't want to bombard you with too much new stuff, too much difficult stuff. I don't want to scare you off year 13 biology. We want to love it, right? So let's skip ahead to these questions, and I'm hoping these are okay for us to do. If you put the question number in the chat and then give me your best answer, I'm going to hang fire on writing my answers until I see some of your answers, and then we'll build up our answers together. Okay. Does it happen in the ribosomes, in the nucleus, or outside of the nucleus? Jennifer, are you talking about the transcription factor bit? Obviously, transcription factors bind to promoter regions, which are on DNA, right? So it's happening in the nucleus prior to transcription, which also happens in the nucleus. So this is all in the nucleus because it's about making the mRNA using RNA polymerase. Okay, so from which biological molecule is a transcription factor made? Fatima, I love how you've given me two options and both are correct. You could say amino acids, or you could say protein. Perfect. Two good options. What is a promoter region? No one's really given me anything yet. Oh, Jasmine says allows RNA polymerase to begin transcription, or yeah, there are some transcription factors that actually inhibit transcription as well. We don't really learn about them at A level. Daisy's saying a specific DNA base sequence at the start of. Can you say anything better than the start of strand? Good, Sire. Good, Silver. So we could say, yeah, it's a sequence of bases in DNA found at the start of a gene. We could also say, I mean, this isn't an exam question, this is just one of my questions, but we could also say transcription factors bind to them. Okay. So, and that will be because of the shape of the transcription factor being complementary to that specific sequence of bases. Will we need to learn about estrogen, progesterone, etc., the menstrual cycle? I don't think the menstrual cycle is on OCR A level, no. The menstrual cycle is not on AQA A level either. I was going to talk to you a little bit about estrogen today, but only because estrogen can initiate transcription as well. It's another example of control of transcription, but I don't think we're going to have time to do that much detail on it. Right, which enzyme is used in transcription and what is its role? Now, there is one enzyme that is more important to mention, right? We mentioned two at the start of the lesson, but what is the important one that's always on the mark scheme? Good. Priscilla, good, says RNA polymerase. And what is its role in transcription? Binds free RNA nucleotides. Ooh, Priscilla. Be a little bit careful there because it sounds like you're telling me that RNA polymerase joins the RNA nucleotides to the DNA nucleotides, which is not the case. Let me do you a little picture. If these are RNA nucleotides, okay, so this is the sugar and the phosphate backbone bit of the nucleotide, and this is the base sticking out. What RNA polymerase does, if I change my pen colour, it joins the RNA nucleotides together, okay, via the sugar and the phosphates. It doesn't join, let me draw some DNA in. If this is my DNA template strand, it doesn't join the RNA nucleotides to the DNA nucleotides. It just joins one RNA nucleotide to the next RNA nucleotide. Okay, so you could say it joins adjacent RNA nucleotides together. Or you could just say it joins RNA nucleotides together, but it's the ones that they are next to. It's not joining the RNA to the DNA. Okay. And what kind of bond? We need to name the bond. So we're going to say by making phosphodiester. Hope you can read that okay. By making phosphodiester bonds. Inc is talking about anticodons joining to codons. So anticodons joining to codons is in translation, and that is just through hydrogen bonding. That does not require an enzyme. The tRNA anticodon, I need 3 fingers. The tRNA anticodon is going to bind to the mRNA codon. That is through hydrogen bonding, and that occurs at the ribosome. So that's translation, not transcription. Question, are hydrogen bonds not formed between the RNA bases and the D? Yes, they are, Saya. Yeah, you will have some weak hydrogen bonds temporarily between those complementary base pairs. Yeah, so you'll have some weak hydrogen bonds forming between the complementary base pairs on the RNA and the DNA. Yeah, but then the enzyme RNA polymerase is joining the RNA nucleotides together. And those hydrogen bonds are only weak, so they will break and the mRNA will move out of the nucleus. How can a transcription factor prevent transcription and gene expression? Now, we haven't really talked about this because in A-level biology, we only learn about the ones that initiate transcription. But just to be aware, there are transcription factors that can block transcription. So what we can say is the transcription factor can bind to the promoter region and stop. Had to think then for a second. Stop RNA polymerase from binding. Okay. It depends on the shape of the transcription factor. And what some of these transcription factors do is they bind to the promoter region and then loads of other proteins bind and form this like big fat protein complex. And then RNA polymerase cannot bind. And guys, if RNA polymerase cannot bind, we're not going to get transcription. Gene expression is switched off. We are not going to make that protein without transcription and translation. Yes, exactly. Silver, it can prevent it by stopping RNA polymerase from binding, so it can't join the nucleotides together, prevents transcription. Can you abbreviate transcription factor to TF? No, I wouldn't. I wouldn't in an exam. With any abbreviation, if you want to abbreviate it, write it out once, then put your abbreviation, and then you can use your abbreviation throughout your answer. But no, I'm just being a little bit lazy. I'm just trying to save time. Don't abbreviate it. I would worry about abbreviating anything. I mean, like RER, SER, smooth, rough endoplasmic, that's usually fine, but I'd still write it out fully just in case, because I'd be like, don't want to lose any marks. Okay. These are harder, but we'll give them a quick go before we finish. Question 1, I wondered whether you could answer this based on year 12 knowledge. This isn't really about transcription, translation. It's just about steroid hormones. We haven't actually learnt about estrogen today, have we? But have a look at the question and see if you've got any idea what you could say for this, because you kind of could answer it from year 12. It's a steroid hormone. It's not made of protein. Steroid hormones are actually made from cholesterol. So why can that hormone enter a cell quickly? Don't worry if you don't know this. We haven't talked about estrogen today. We didn't have time. But they can enter cells quickly by passing through the cell surface. What's the cell surface membrane made of, guys? Doesn't need a channel protein. Doesn't need a carrier protein. What's the cell surface made from? Cell surface membrane. What's it made from? Good. And estrogen is a steroid hormone. It's not made of protein. It's made from cholesterol, which is a type of lipid. And there is cholesterol in that bilayer. Yeah, cholesterol is lipid soluble. So estrogen, because it's a steroid hormone and not a protein, is lipid soluble, which means it can simply diffuse through the phospholipid bilayer. Because if it's lipid soluble, you may remember from year 12, if it's lipid soluble, it can diffuse through the phospholipid bilayer, and estrogen is lipid soluble, basically. If it's not lipid soluble, if it's water soluble or polar, it can't diffuse through the phospholipid bilayer. It has to use a channel protein or something. But this hormone can simply diffuse. Now, you might be thinking, hang on a minute, why are we talking about estrogen when we've just done a lesson on transcription and translation? It's just because estrogen is a hormone that does affect transcription. So sometimes it comes up in this topic, okay? Don't worry about it today. But estrogen can actually initiate transcription in a cell because it diffuses through the phospholipid bilayer, and it binds to its receptor, and then it actually can initiate transcription. It can move into the nucleus and it can initiate transcription. That's the only reason this question has come up, okay? Aren't hormones proteins? Not all of them, no. You'll learn about that in year 13, okay? A lot of hormones are proteins, but some, like estrogen and testosterone, are what we call steroid hormones, okay? They are made from cholesterol and they are lipid soluble, okay? So they can just simply diffuse into the cells, and their receptors are inside the cytoplasm, not on the membrane. But you will do more about that, don't worry. And just to finish then, in the cytoplasm, say this is another, testosterone is another steroid hormone. It's another lipid soluble hormone. Testosterone binds to a specific receptor called the androgen receptor. This receptor is made from protein. Explain why testosterone binds to a specific receptor. This is year 12 knowledge, guys. I know it might not seem like it, but it's about shapes fitting together. We're explaining why a particular hormone binds to a particular receptor. It's just about shapes fitting together, okay? You don't really have to use any year 13 knowledge for this. Okay, so they are complementary. Complementary, right. Jasmine, I was hoping someone would say active site. Why are we not going to talk about active sites here? Why are we not going to talk about substrates and active sites? We've got a specific receptor. The receptor is made from protein. Exactly, Jay. It's not an enzyme. Okay, it's so hard. It's like everyone falls into that trap. As soon as you think about proteins and shapes fitting together, you automatically assume enzymes and you talk about active sites. This is a receptor. It's not an enzyme, but it is a protein. So the receptor has, we could say it has a specific tertiary structure because it is a protein, but we're not going to say it's got an active site, are we? Okay. Yeah, you're on to me. So we could just say the receptor as a protein has a specific tertiary structure, and we could say, we can still use the word complementary in shape to testosterone. Okay, so testosterone is a steroid hormone, but it will have a specific shape, right? And that shape is going to be complementary to that receptor because that receptor has a specific tertiary structure. I really did just include that question just to see if you would fall into my trap of talking about active sites. So that was my fault. Okay. Right, that's all we've got time for today. I know we've done a few different bits and bobs here. Like we've done we've mentioned hormones, we've talked about proteins, we've done transcription, translation. The new thing that we've learned is transcription factors, right? So what I'd like you to take away with you is that a transcription factor is made from protein. It binds to a promoter region and it can initiate transcription, okay? Even if you can't remember that, I'm still hoping our revision on transcription and translation has been useful, okay? Giving you a little bit more confidence. Right, I'm going to let you guys go. I've kept you 4 minutes too long. How long is the homework available? I'm actually not sure. I'm guessing it'll be till the end of August. Yeah, we haven't done the estrogen bit, although I'm not being funny, but this question, we haven't done this today, right? But this question is a use the diagram question. So you could have a go at this type of question if you want to, because the answers are kind of on the diagram. So you could have a go at that. But yeah, if there's anything on the homework that you can't do, just miss it out. You know, it's just to have a go at, right? So if there's any questions you really can't do, miss them out or just have a go. Thank you so much. I will see you next Thursday. Have a lovely weekend. Do some biology revision or not. Yeah, have a great weekend, guys. Would we ever go over the stuff we missed? I wasn't gonna go over this next lesson. I mean, if you look at this one, if you want to stick with me, who is it? Is it L? If you want to stick with me, this was about estrogen. You can go if you want to, guys. I don't mind. I won't be offended. But if you want to stick with me, this question was about a hormone like estrogen. Can you see how it's diffusing? It goes straight through the phospholipid bilayer. Okay, because it's a steroid hormone, it's lipid soluble. So on this diagram, you can see it's going straight through the plasma membrane. It's diffusing through. Now have a look at what it's doing, guys. You can see it on the diagram. This hormone is binding to an inactive transcription factor. And then look, when it binds, it activates the transcription factor. You can pretty much see all of this on the diagram. Then look what's happening. The transcription factor is binding to the start of a gene. So A on here is our promoter region. And then look what's happening. When it binds to the promoter region, RNA polymerase binds. So it's initiating transcription. But you can kind of get all that from the diagram. So estrogen works in this way. It diffuses into the cell through the phospholipid bilayer. It binds to the estrogen receptor, which is on a transcription factor. It activates the transcription factor, which then binds to the promoter region, which then initiates transcription. Okay? Because yeah, as you've just said, Inc., the transcription factor, when it binds, it's allowing RNA polymerase to bind. That's going to initiate transcription. So estrogen is initiating transcription. It's a hormone that can control transcription. Okay, that was really all we missed. Because you've got the same diagram again here. So if you want to learn a little bit more, have a little read through that diagram, which explains it in a lot of detail. And then have a go at this question using the diagram, just explain the steps that you can see. Okay, right, I'm gonna go. I hope that was helpful. I don't know who's still here. Maybe I'm talking to myself, but yeah. Some transcription factors do stop transcription. Yeah, we don't really learn about any of them in A level though, Saya. But this one is activating transcription because the diagram's trying to show you, I think here, is trying to show you that when the transcription factor binds, RNA polymerase binds. Thanks, guys. Right, I'll see you next Thursday. Have a good one. Have a good weekend. Thank you.